be back, guys. In this video we are going to look at translation in bacteria, and is being presented by your girl Ruth, the .com scientist. In the previous video, in this very series of bacteriogenomics, genetics, we've looked at transcription, we've looked at regulation of transcription. Now we are going to look at translation. At the end of the day, all these processes take place because we need to get proteins, which proteins are very key to the survival of the bacteria in different ways. So in this specific video, you're expected to learn and memorize the different steps which take place during translation in the protein synthesis pathway. Okay, so we are going to look at what happens next. We ended at looking at messenger RNA being formed. So what happens next is going to take place in the ribosomes. The ribosomes are the proteins, are the organelles which are going they're like the machinery for making proteins. The information for the protein making has been brought by the messenger RNA. Now it is up to the ribosomes to get us the proteins that we need. The ribosomes don't do this alone. They require transfer RNA. They require very many factors, as we are going to see in this video. So what happens next is that on this messenger RNA which has been formed, we have a sequence, a special sequence called the Shine-Delgado sequence. This sequence is called the Shine-Delgado sequence. So what does this sequence, what is its significance in this process? The Shine-Delgado sequence is GGA, GGU, GAGU. If you don't want to forget, GAGU, but make sure the GA is double G. Now this sequence is found on the messenger RNA, the GGA, GGU. It's the Shine-Delgado sequence. It's found on the messenger RNA. Let's not forget that. Now, let's also remember that these ribosomes, they have two subunits. They have the 30S subunit and they have the 16S subunit. The 30S subunit is the one which binds to this sequence. It's going to bind to this sequence, the Shine-Delgado. How does it bind? It binds because the 16S subunit contains a complementary sequence. Complementary sequence to this is ACC, UCC. This is backwards because, as you know, A binds to U. So it binds to that. Now imagine your RNA is there swimming around and it meets this sequence. It's going to bind to the ribosomes. So this is going to be the start of our protein manufacturing. This binding requires to be stabilized, and the stabilization of this binding is going to be done by initiation factor 3 with the help of initiation factor 1. Now, where is this Shine-Delgado sequence located? If you see that the Shine-Delgado sequence is about 4 to 6 nucleotides from the initiation codon. This is where it's located, about 4 to 6 nucleotides from the initiation codon. Now, we've talked about transfer RNA and the fact that transfer RNA is going to help us transfer RNA is going to carry the different amino acids. And we should also be noted that methionine is the first amino acid which is carried to the ribosomes during translation. The first transfer RNA, which is carrying the first amino acid, binds to this complex. The complex we are talking about is the combination of the ribosomal RNA and messenger RNA with the aid of initiation factor 2. We see these initiation factors having a key role in stabilizing and in making some of these processes happen. When this happens, there is that pairing. The anticodon USC, which is usually the start codon, is going to bind to the complementary codon, which is AUG. Okay, so let's have a brief look into the ribosome. The ribosome is containing three key sites, three important sites. One of the sites is the peptidyl site, or it can also be abbreviated as the P site. This site contains the transfer RNA. It's going to have our transfer RNA, which is carrying our methionine. It is why the P site is the peptidyl site, because that is the first transfer RNA to come and enter the ribosomes. We shall see how it happens in the coming slides. You would have to see the picture. So what happens is, after this transfer RNA has entered into the P site, the adjacent site is the A site, acceptor site. You can just remember it from acceptor, A site, A, acceptor site. Then P is the peptidyl site. We shall also look at the other site as we go ahead, the exit site, which is E. So when another transfer RNA brings the second amino acid, it's going to occupy the acceptor site. When it occupies the acceptor site, the methionine, which is the amino acid, is going to form a bond, a peptide bond with the second amino acid which has been carried. We've looked at these peptide bonds and how they are formed between the amino acids. So that bond is going to be formed. So that is roughly what happens during elongation, and they'll keep moving. So this is what I was talking about. We have the A site, which is the acceptor site. We have the P site, the peptidyl site, and we have the E site. The first transfer RNA, which is carrying methionine, is going to come and sit in this site. When it sits here, the second transfer RNA, which is going to come, is going to sit here. So when they sit here, when it sits here, if you can see down here, you find that these two amino acids, which are close to each other, they are going to form a peptide bond. And the moment they form a peptide bond, the messenger RNA is going to move. It's going to move along the ribosomes, and before you know it, the transfer RNA, which is here, is going to be in the exit site. Meaning, the one which was in the acceptor site is going to now be in this site, the peptidyl site. Meaning the acceptor site is going to be empty at the moment. It will be very ready to receive another transfer RNA. So basically that is what happens. And this is why this is more like the elongation process, the elongation step. So let's look at the details of exactly what happens. These things don't just move de novo. They are activated by something. So basically what happens is the amino acid tRNA, the transfer RNA which has the amino acid, it first binds to a complex elongation factor. Elongation factor, this is the elongation factor TU, and the GTP prior to the A site. So this is going to bind with the elongation factor, and when when they bind, the elongation factor is going to place the amino acid TNA into the A site. So we've seen that the amino acid TNA does not just come and enter the acceptor site. It is placed there by something, and this is what places it there. It is the elongation factor TU. When that happens, GTP is going to be hydrolyzed to GDP plus a phosphate group. So that is what happens. Meaning there is hydrolysis, and which means that there is energy required for this to happen. Okay, so now it has been placed into the acceptor site. So we see that now the elongation factor TU, when it combines with GTP, this complex is released, and a elongation factor TS releases GTP from EF, or elongation factor TU, and reloads it with a fresh GTP. So we talked about formation of the peptide bond between the two amino acids. This is what happens. This is how it happens. There is an enzyme called Peptidyl Transferase. This enzyme, its activity is done which leads to the formation of the peptide bond between the two amino acids. So when that happens, it means the peptide bond has been formed, and therefore the subsequent amino acids can be brought in to form our polypeptide chain. So there is translocation. We talked about movement. We need the amino acid, we need the transfer RNA to move. The one which has already bound to our peptide bond has to get into the exit site, such that you can have a free site for a new transfer RNA. So translocation is the final step when the ribosome moves 3 nucleotides. The ribosome is one which moves, let's take note of this, it is not the messenger RNA, it is the ribosome. It moves 3 nucleotides, and the free transfer RNA enters the E site. So it is free because it has already given us, its amino acid has already been used, it has already taken part in the formation of the peptide bond, meaning it's now free. We need to release it such that we can get another one. So this process continues until we form our peptide bond. So termination. As we looked at transcription, there has to be some form of termination of this process. So termination during this process of translation, it occurs when a termination codon occupies the S site. The termination codon, we are going to look at it, and we need to keep these termination codons in mind, because when you're describing this process and you talk about termination and you don't talk about them, then it is not complete. You need to give us, you need to really state the termination codons. For this case, it is UAA and UAG. So this termination, the ribosomes are going to move along the messenger RNA until they meet the termination codons. The termination codons are going to be recognized by release factor. Now we are looking at release factors. We've looked at elongation factors. We even have these the release factors. So these factors, you need to keep them in mind. You need to remember them when you come to describing this process. The release factor 2 is the one which recognizes UAA. We need to take note of the factors, the enzymes that are involved, such that you can really get this process. So this is like a summary of what we've talked about. We've seen that we have the ribosome, we have the larger subsection, we have the smaller subunit of our ribosomes, and we have the messenger RNA which has been formed. We have the transfer RNA. So basically, this is what we need. This is what we need to make a protein. The first step, which is the initiation stage, we see that the first they first have to form a complex. First of all, the messenger RNA recognizes it has the shiny organo sequence. So I'm trying to give a brief summary of what we've talked about. So when it is recognized, the ribosomes are going to bind to the messenger RNA. So we shall have our complex formed, the initiation complex. So when that happens, we shall see that we are going to see that the first transfer RNA is going to bring our amino acid, and it is always methionine. Don't forget that this transfer RNA is going to, it enters the peptidyl site. It doesn't enter the acceptor site. It enters the peptidyl site. So when that happens, they are going to bring different amino acids. They are going to be brought by the different transfer RNA, and peptide bonds are going to keep being formed. And then the transfer RNA which is no longer carrying an amino acid is going to be released. A new one is going to join, and then we have the termination. Here, the release factor recognizes the stop codon, and the moment it's recognized, there is no more formation. There is no more transfer RNA bringing amino acids. Therefore, as you can see, the release factor is going to be occupying the acceptor site. Therefore, no more transfer RNA can bind to our acceptor site. So this is basically what happens. Thank you for taking time to read. I hope you understood something. If you didn't, please leave a question. I'm going to try and get a video that can animate this process, such that you can get an easier grasp of the content. Thank you. Subscribe if you like this. Give it a thumbs up, and then hit the notification bell to be notified whenever I post. Thank you so much, guys. All the best in your endeavors. All the best in your reading. Bye-bye.